All right, today we're going to be molecular detectives. We are diving deep into the amazing science of immunoassays. That's the powerful technique that's really behind so much of modern medicine. Let's jump right in.
So just think about this challenge for a second. How can we find just one specific molecule among trillions, literally trillions, inside the human body? Your body is this bustling metropolis of molecules. Trying to find just one type, like a hormone or maybe a tiny piece of a virus, it's like trying to find one specific person in a city of billions of people and you don't even have a photo. Well, that's where immunoassays come in. They're the tool that makes this seemingly impossible task, well, possible.
And this isn't some super abstract science that only exists in a lab. Chances are you've actually held an immunoassay in your own hands. Ever seen a home pregnancy test? Yep, that's one. This exact same technology is used for all sorts of things: screening for drugs, detecting infectious diseases, and even finding those super early markers for cancer. It's everywhere, and it's been quietly changing the game for years. Okay, so here's how we're going to break it all down. We'll start with the detective work idea, then meet our key players, the antigen and the antibody. We'll get into something called the Goldilocks reaction, then figure out how to make the invisible visible. After that, we'll look at two main ways to play, and finally, we'll talk about the incredible power of these tests.
So yeah, at its very core, an immunoassay is really all about biological detective work. The mission, if you choose to accept it, is simple: find and then measure a very specific target molecule inside a really complex sample, like blood or urine. And to pull this off, our detective needs two things: a target, or a suspect, and a detector. In the immunoassay world, we call these the antigen and the antibody. And you've got to understand, their relationship is incredibly specific. We're talking a perfect lock-in key. So, let's meet our detector, the antibody. It's this cool Y-shaped protein, also called an immunoglobulin. And honestly, you can just think of it as a microscopic heat-seeking missile. Its real power is right there at the tips of that Y. They're shaped to bind to one, and only one, specific target. This insane level of specificity, well, that's the secret sauce to this whole thing. Okay, so let's make this crystal clear. The antigen, that's our target. It's what we're searching for. It could be a hormone like the one in a pregnancy test, or maybe a protein from a virus. And the antibody, that's our detector. It's the how. It's engineered to find and latch on to a very specific spot on its matching antigen, and totally ignore everything else around it.
Now, it's not enough to just have antigens and antibodies floating around. For the test to actually work, we need them to bind together and form these big measurable clumps, or complexes. And for that to happen, the conditions have to be, well, like the fairy tale says, they have to be just right.
So imagine this. You've got just a few targets, our antigens, but you flood the system with a ton of detectors, the antibodies. What happens? Each little target gets totally mobbed by individual antibodies. There's no room for them to link up, for one antibody to grab a target and another antibody to grab that same target to build a chain. So the result? A really weak, pretty much unmeasurable signal. Okay, so what if we flip that? What if you have way too many targets and not nearly enough detectors? Well, every single binding site on every antibody gets clogged up instantly by a different target molecule. Once again, you can't build those cross-linked chains. And just like before, you end up with a weak signal.
But when that ratio is just right, in what scientists call the equivalent zone, that's when the magic happens. You've got enough antibodies to link multiple antigens, and you have enough antigens to be linked by multiple antibodies. It creates this big, stable, almost lattice-like structure. And that is what gives us the strong, clear, measurable signal we're looking for.
And this graph here, it shows you exactly what I'm talking about. This is a classic precipitant curve. See over on the left? That's when you have too many antibodies, low signal. And way over on the right, too many antigens, again, low signal. But right there in the middle, that sweet spot, that's the equivalent zone, where the signal just peaks. That is our perfect Goldilocks moment.
Okay, so we've solved one big problem: getting our targets and detectors to link up in a way that should be measurable. But there's another, even bigger problem. All of this stuff is happening at a molecular level. It's totally, completely invisible to the naked eye.
Right? So now what? Even if you get that perfect binding, even if these big complexes are forming, how in the world do you actually see it? You can't just squint harder and count the molecules. We need a trick. We need a way to make this invisible event visible.
And the solution is actually pretty genius. We attach something called a label to our antibody. Just think of it like a tiny signal flare. It's a molecule that, when we tell it to, produces a signal we can see. Maybe a change in color, a little flash of light, or a fluorescent glow. It could be an enzyme that causes a color change, for example. In fact, this label is exactly what makes that second line on a pregnancy test turn pink.
So now we have all the pieces of our puzzle, right? We have the antigen, the antibody, and the label. And we can put them all together using two main strategies. Let's take a look at how they work. First step, you have what's called a competitive assay. In this one, you've got the antigens from your sample competing against a bunch of pre-made, labeled antigens for a limited number of spots on the antibodies. So think about it. The more of your target antigen there is, the more it wins that competition, which means you actually get a weaker signal. It's a little backwards, right? More target means less signal. Then you have the other super common method, the non-competitive or sandwich assay. This is way more straightforward. One antibody grabs the antigen, and then a second labeled antibody comes in and grabs it too, making a little antigen sandwich. In this case, it's simple. The more antigen you have, the more sandwiches you make, and the stronger the signal gets.
So after all this clever molecular biology, what's the payoff? Well, it gives us the ability to detect molecules with a level of sensitivity that is, honestly, it's hard to even wrap your head around.
I mean, look at this number. The precision is just mind-boggling. Modern immunoassays can get down to a detection limit of 10 femtomoles per liter. So what does that even mean? Well, to put it in perspective, that's sensitive enough to find about 600,000 specific molecules inside a single drop of a sample. That is just breathtakingly precise.
And believe it or not, it gets even more amazing than that. New techniques like digital ELISA are pushing this to the absolute limit. What they do is they separate every single antibody complex into its own tiny microscopic well. We're talking hundreds of thousands of them. Each well is so small it can only hold one, if any. Then the machine just lights up the wells that have a target inside, so it can literally count individual molecules. It's the difference between guessing how many people are in a crowd and doing a perfect one-by-one head count. And this incredible power is why immunoassays are so important. This is what lets us reliably diagnose a pregnancy from just a tiny amount of hormone. It's how we can screen for drugs so quickly, detect viruses like HIV, and, this is huge, find the markers for diseases like cancer at their absolute earliest, most treatable stages. All right, so let's just quickly recap the big ideas here. Antibodies are nature's super-specific binders. Antigens are the targets they're looking for. Getting that Goldilocks ratio between them is absolutely key for a strong signal. We use labels as little signal flares to make the whole invisible process visible. And all of this together, it powers the diagnostics that we all rely on, from the simplest tests to the most revolutionary ones. Which, of course, leaves us with a pretty fascinating question for the future. As this technology gets more and more sensitive and gives us the power to find almost any molecule you can think of, what are we going to be able to diagnose next? What currently invisible signs of disease are we going to be able to spot long before they ever become a real problem?